KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, Xylem, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Education Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Okay students, now in this video we are discussing complex tissues information. So in complex tissues, let us start to learn the xylem. Why? Because so our knowledge point of view, complex tissues, so two types of complex tissues are there now. So what are those? One is xylem and second one is phloem. So these two members we will be calling as complex tissues. So these two members means what are those? Xylem and phloem, which are present in the vascular bundles. So that's why these two members we will be calling as vascular tissues. So next one, what are the group of the plants consist of these vascular tissues? Now all those plants we will be calling as tracheophytes, isn't it? Tracheophytes means the plants which consist of both xylem and phloem we will be calling as tracheophytes. Now their xylem members point of view. Okay, xylem, so definitely which conducts water and minerals. So which conducts water and minerals from roots to aerial parts of the plant body, isn't it? So there now if you see this particular structure, this particular xylem, our convenience to study, now we have been divided into four types. First one is tracheids, second one is vessels, third one is xylem fibers, and fourth one is xylem parenchyma. One more time, let us learn. So four types of xylem members. One more time: tracheids, vessels, xylem fibers, and xylem parenchyma. Out of four types, so this particular point in yesterday itself we learned. Out of four types, three are dead. First three, tracheids, vessels, and xylem fibers are dead, and xylem parenchyma is living tissue. One important point we learned in a sclerenchyma, what is that point? No, suppose if the cells are living means definitely cell wall is made up of cellulose, and living means definitely within the cell protoplasm or the protoplast is present. Protoplasm is present. Now these 2 points we learned already in previous videos. Suppose if you didn't hear means at least now you try to learn. So what is the differentiation or what is the difference between dead cell and living cell means dead cells cell wall is made up of lignin, and the living cells cell wall is made up of cellulose. Dead cell ulle cytoplasm or the protoplasm will not be there, whereas in a living cell within the cell we can observe the presence of protoplasm. My point you understood now. So thereby these two are main differences finally. Okay, now let us start to discuss tracheids. Now tracheids are elongated tube like cells. One more time, tracheids are elongated tube like cells are there, tube like cells with thick and lignified walls. Once you see that, so when we will be observing the presence of lignified cell wall, what is the point mainly we have to understand? These particular cells are dead. So that is a significant point now we learn. Okay, lignified cell walls are there and with tapering ends. Once I have given in fiber tapering ends I shown no. So tapering ends means very pointed structures madri irk. Tapering ends. So definitely for this one in the next video I will give diagrams. At the time you will understand clearly. So both tracheids diagram I will give as well as vessels also I will give. So no worry. Okay, right. Now tapering ends are there, and these are dead and are without protoplasm. So means here the cells are lignified means that time only we learn definitely these cells or these tracheates or cells are dead we seen, and dead means definitely which one is absent? Protoplasm is absent. Okay, next one, the inner layers. The inner layers of the cell walls have the inner layers of the cell walls have thickenings. So the inner layers of the cell walls have thickenings which vary in the form. So means thickenings means the distribution of the the distribution of the lignin will be in dissimilar. So means here in one place that will be very thick, and in some other places the thickening of lignified walls will be changes. So likewise the thickenings will vary in form. So thereby next point is in flowering plants. So if you take these particular flowering plants, what is the main important point? The tracheids and the vessels are main main water transporting elements. So suppose if you see that there are 4 different types of elements are there in xylem: tracheids, vessels, xylem fibers and parenchyma. So there out of 4 types, first 2 means what are those? Tracheids and vessels definitely or mainly helps in conduction of water and minerals. Where? In flowering plants. Don't forget, in flowering plants only tracheids and vessels are main water transporting elements. So these are all points we can see related to the tracheids. Next one, vessels point of view. What are the points we can be discussed regarding the vessels? Means vessels or vessel is a long cylindrical tube-like structures made up of with many cells. Did you understand? So thereby here this particular vessels also, it is also being like how long cylindrical tube-like structures made up of with many cells. Made up of with many cells. That particular cells we will be calling as vessel members. Vessel members. Once let me give you one important point. What is the point? In comparison of tracheids and vessels, in comparison of tracheids and vessels, suppose if they ask which one is more developed means, so we can find out later one means the second one only more developed than the previous one. Okay, so thereby in tracheids and vessels more developed one is vessels. Again, you don't compare with xylem fibers and xylem parenchyma. When you compare to the all xylem parenchyma more advanced, you don't think likewise. Okay, so in comparison of tracheids and vessels, most advanced one is vessels. So next one, these particular vessels are the made up of with so many cells or the many cells known as vessel members. Vessel members. Next one, each with lignified walls. So definitely each vessel cell, vessel members definitely lignified walls. Why lignified wall? Definitely that particular cells are dead. That is only we learn. So next one, with large central cavity. So which one is absent? Protoplasm is absent. So thereby empty space will be there. So thereby we can find out large central cavity. Large central cavity we can find out. Don't forget, the presence of large central cavity is the best important point to remember the vessel members. Whereas in tracheids, if you see there in this tracheids, so here thick lignified walls are there because of the thick lignified walls. So here in tracheids empty space is narrow. This particular point you try to remember. Okay, so in tracheids, because of the more thickness and lignified walls and with tapering ends, empty space is narrow. Where in tracheids? Point on understanding now. So thereby next in vessels, lignified walls are there. Lignified walls are there, but even though lignified walls are there, when compared to the tracheids, very thin cell walls we can see in case of vessels. So that's why here we can find out large central cavity. Central cavity is more than the tracheids. Okay, so this in previous papers I have seen this bit, that's why here giving to you. So in vessels, a large central cavity is present, means here the empty space within the cells or the vessels will be more than the tracheids. This particular point you try to remember, like a difference point of view we can differentiate. Next one, vessel cells are also devoid of protoplasm. Means here the cell walls or the vessel cells are devoid of protoplasm, means here we are observing very large central cavity. Why we are finding large central cavity? So because of the absence of protoplasm. If protoplasm liquid is not there, means cytoplasm is not there, means at the time definitely we can find out empty space. That particular empty space is more in case of vessels. Getting only now. Next one, vessel members are interconnected. The vessel members are interconnected through the perforations. Means here, what are the vessel members are there? If only one vessel member will not be there, LMA together will be there. So all vessel members together will be there. No. So at the time when all vessel members are beside is there. No. At the time all cells are connected through the perforations. Perforations are holes. So in a fiber, one diagram I have drawn in previous videos. No? So likewise here also vessel members. Vessel members how it will be. Once you see there long cylindrical tube-like structures, no? Like tube-like structures, once you think. Okay, once you try to think in your mind. So thereby, suppose one cell definitely particularly consists of hole-like structures, means because of the presence of hole-like structures, all cells are connected each other. I think so you are getting, but no worry, in the next video definitely I will give one suitable diagram, so thereby you will understand. Okay, no worry. Next one, now here what is the point now? Vessel members are interconnected through perforations in their common walls. Next one, the presence of vessels is a characteristic feature of angiosperms. This 100 times underline pannikonga. Okay, so means here the presence of vessels is a characteristic feature of angiosperms. Okay, means here what is the main important point? Now only we are learning the presence of vessels. Presence of vessels is a characteristic point of angiosperms. So don't forget, very important. Next one, what are the main important points from one more time? Next one, gymnosperms lack the vessels in their xylem. So here that particular point here I didn't give you, but here what is the point now I am giving through my voice. No? So the presence of vessels is a characteristic of angiosperms, and the absence of vessels is a characteristic point of gymnosperms. Means in gymnosperms which one is absent? Vessels. In the gymnosperms, vessels. So the presence of vessels is a characteristic feature of angiosperms. Likewise, in gymnosperms, once you see that gymnosperms lack vessels in the xylem. Now, presence of vessels is a characteristic feature of angiosperms, as well as absence of vessels. Lack means absent only. No. Absence of vessels is a characteristic point of gymnosperms. Saving only. No. Now, out of 4 types, tracheids, vessels, xylem parenchyma, xylem fibers and parenchyma. So these two we have completed. Here what is the main important point we are discussing in vessels? Means in vessels, what is the point? No, presence of vessels we are observing, or the presence of vessels is a characteristic feature of angiosperms, and absence of vessels is a characteristic feature of gymnosperms. Don't forget, save in your mind. Next one, third one, xylem fibers. Now, xylem fibers point of view, xylem fibers are highly thickened walls. Don't forget, highly thickened. Again, in dead cells, highly thickened means only because of the presence of lignified cell walls. Highly thickened means in a dead cells, wherever you see in our botany subject point of view, if we are finding thick thick walled means thick walled and dead means that is only because of the lignin. Right. Next one, obliterated central lumens. Obliterated central lumen. This particular word. So suppose if they would like to ask any kind of questions related to the xylem fibers. So xylem fibers, just one word you try to remember, that is obliterated. Obliterated. Suppose I told you already, without mentioning of obliterated, they cannot ask the question under xylem fibers. So this much important xylem fiber. Why? Because here once you see there, you are highly lignified or highly thickened cell walls are there, no? At the time what we can find out means obliterated central lumen we can see, means there like central lumen, very thin central lumen we can find. Yeah, ma'am. Because suppose more thickness is there means central lumen is there, no? That particular central lumen size will be decreased. If highly thickened cell walls are there, no? Because of this nature, empty space will be very less. That one we will be taking as obliterated central lumen. Okay, sir. Next one, they may either be septate or aseptate. Septate or aseptate means once I have given in a fungal members, septa means so suppose one particular cell is there means particular cell suppose if you are observing separate layer or the separate wall, so such particular layer or the line we can be taken as septate, or suppose not there means that particular separating wall we will be taking as aseptate. A indicates absence, no, aseptate. Right. But my dear students, in xylem fibers, one and only the word you try to remember, that is obliterated central lumen is related to the point of xylem fibers. Next one, xylem parenchyma. Xylem parenchyma point of view, xylem parenchyma cells are living. This is the interesting point. Why? Because till now 3 members we have discussed under xylem, 3 are dead. Now in xylem one and only the living, that is xylem parenchyma. So here the xylem parenchyma cells are living, thin walled, and their cells are made up of cellulose. So here the living cells means definitely I told you the cell walls are made up of cellulose. Next one here, they store which one? Food materials in the form of starch or fats or other substances like tannins. So but definitely helps for storage. So that particular point mattum try to say either starch or fat or other substances or whatever it is, but useful for storage purpose only, no? So that's why definitely this particular xylem parenchyma helps for storage. Storage of food materials. Next one, the radial conduction of water takes place by the ray parenchymatous cells. So here the radial conduction. So here the movement of water in radial conduction. So definitely that is under the control of ray parenchymatous cells. Ray parenchymatous cells. So what is radial and what is ray parenchymatous? In the next videos definitely I will explain. So right, next one, now let us see. Now the primary xylem is of 2 types. If xylem is there, no, there xylem, primary xylem. So that particular xylem is of 2 types. So there, what are the 2 types means? So first one is protoxylem and the second one is metaxylem. Simply primary xylem is divided into 2 types. One is protoxylem and the second one is metaxylem. If protoxylem means what and metaxylem means what? Now let us see. So there, now first formed primary xylem, first formed primary xylem is called protoxylem. Now paringa, first one proto, second one meta. Our subject point of view, suppose if any kind of structure formed first means, so such kind of structure we can be taken as a pro. Even in mobiles also, no, pro they will use. Pro, suppose if they are using means adi ange mobile ange model only first one, that is the meaning. Okay, so now you see there the first formed protoxylem, or sorry, the first formed primary xylem elements are protoxylem. Why? Because here we are calling protoxylem because of first formed one. First formed primary xylem is protoxylem, and later formed appadina not first time, second time madri. So later formed one is later formed primary xylem is metaxylem. So thereby, so here we have seen, no, primary xylem is of 2 types. One is protoxylem and second one is metaxylem. So adi one type. Now we are entering into the most important concept that you listen carefully. Now in stems, protoxylem lies towards the center or pitta. Okay, just sentence mattum try to remember, or I'll teach you, just let us discuss, let us say this one 2 or 3 times, so thereby it will store in our mind. In stem point of view, protoxylem lies towards the center and metaxylem lies towards the periphery. So that kind of condition we will be calling as endark type. Or simple point amma. So till now what we read, no, that you forget, but this point you try to say what is that point. No, in stem protoxylem situated towards the center and metaxylem towards the periphery of the organs. So such kind of condition we will be calling as endark type. Next one, in roots protoxylem lies towards the periphery and metaxylem lies towards the center. Once you see there stem and root information, once you see in stem, no, protoxylem it is situated towards the pitta and metaxylem which is situated towards the periphery. So such kind of arrangement of xylem members we will be calling as endark. Ipu root paringa, root exactly situated opposite to the stem, you see there. If protoxylem it is lies towards the where periphery and metaxylem it is situated towards the center. So roots arrangement is opposite to the stem arrangement, or stem arrangement is opposite to the roots arrangement, vice versa. So that's why in roots, no, arrangement of xylem is exarch condition, whereas in stem, no, endark condition. Endark and exarch. Just words you try to remember. When I will start the information of anatomy, definitely I will give you information regarding this one, no worry. So what are the topics or what are the new words of you people are listening, no? So each and every word explanation definitely diagrammatically I will explain. Right. Now suppose if you are there in front of me, means on the spot chance is there to write and to draw and explain. Okay, ma? So you try to understand the teacher's feelings also, right? Next one, so this is the information regarding xylem. Okay, next one, phloem point of view, what is the information to be discussed related to the phloem? So definitely this particular phloem members transports of food materials usually from leaves to other parts of the plant. So phloem members point of view, definitely phloem transports the food materials usually from leaves to other parts of the plant. So thereby once we see, no, suppose if you take xylem members that transport the food materials that transports the water and minerals only in unidirectional flow, likewise we have seen in xylem. Likewise in phloem, food materials conduction in 2 ways. One is in bidirectional or in multidirectional. Bi or multidirectional movement of food materials we can see, but water molecules conduct only in unidirectional. So that particular point also you try to say. Right. Now here, now let us point next point. Phloem in angiosperms is composed of, so in phloem in angiosperms, no, phloem members, how many parts we can be taken now? So composed of sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Reverse, so what is the point is reverse, no? Our knowledge point of view, how many are dead over there in xylem? How many dead over there in xylem? 3, no? So like in this particular case, in a phloem, no, 3 are living will be there and 1 will be dead will be there, where in case of a phloem. That's why phloem structure phloem's members is reverse to the xylem members. What is the point now I am giving you in your mind means, in xylem members of 4 types are there. Out of 4 types, 3 are dead, 1 living we see. Now what is the point we are observing, we are learning means in phloem, out of 4 types, 3 are living, 1 is dead. So what are those? No, once let us see. Sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Can you guess out of these 4 types which one is dead? Last one, phloem fibers. Fibers always dead. Don't forget, fibers always dead. That's why phloem fibers. So these phloem fibers we can be taken by one more name, bast fibers. So next video definitely I will give you that particular point. So this particular point meaning bast fibers. So that particular one is bast fiber. Now information, now let us see. Okay, now very important point, first point only. Gymnosperms have, angiosperms point of view, in angiosperms, monocots and dicots. Angiosperms means in angiosperms, no, 4 types of xylem member or phloem members are there. Sieve tube elements, companion cells, phloem parenchyma and phloem fibers are there, no. So now here in gymnosperms, after gymnosperms, angiosperms are formed now. Means when compared to the gymnosperms, angiosperms only more advanced. So now here in gymnosperms, what are the points? Gymnosperms have albuminous cells and sieve cells. Over 100 times by heart panninga. So whether you will by heart or not, whatever it may be, that mode you try to follow, but these particular points we should not forget. In angiosperms, sieve tube elements are there, isn't it? Sieve tube elements are there in angiosperms, but in gymnosperms, sieve cells are there. The difference is in angiosperms, sieve tube elements, but in gymnosperms, sieve cells, don't forget. Okay, along with the sieve cells, one more is albuminous cells. Albuminous cells and sieve cells are present in gymnosperms. And so here, that's why here I have given one point. They lack sieve tube elements and companion cells. Means in gymnosperms, sieve tube elements are absent as well as companion cells also absent. So instead of companion cells and sieve tube elements, in gymnosperms, what are the structures we are observing? Albuminous cells and sieve cells. Important. Next one, first one, let us start to discuss sieve tube elements. Now these sieve tube elements are also long tube-like structures. Now first of all, in tracheids and vessels, likewise only we seen sieve tube. Tube comes means tube-like structures. So are long and tube-like structures arranged longitudinally and are associated with companion cells. Once you come up here, you see there sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Line wise one sequential order is there now. Here also arrangement also will be like that only. Sieve tube elements definitely connecting with the associated with the companion cells. If sieve tube elements and companion cells are present together or associated together. This particular point important. Why sir? If sieve tube elements and companion cells are associated together means one important point to be discussed. So that point let us discuss now. Their end walls are perforated. Perforated means what is the point we have to remember? Holes. End walls, no, definitely what we can find out? Holes we can observe. Holes we can observe in sieve-like manner to form sieve plates. Sieve plates means suppose mummy for cleaning of a floor, no, she will use one sieve plate. Likewise here also in end walls of the sieve tube elements, what we can find out? Sieve plates we can find out. Sieve plates we can find out. Next one. A matured sieve element possesses a peripheral cytoplasm and a large vacuole but lack of nucleus. My dear student, one point you try to say, suppose if you take matured one, matured one or matured sieve tube element, suppose if you take there peripheral cytoplasm. Peripheral means cytoplasm is situated towards the walls or towards the ends. Towards the end, and a large vacuole also is present. Large vacuole is present, but which one is absent? Lax means absent, and which one is absent? Nucleus. My dear students, here what is the point? No, if you take a young one, here mature sieve tube elements, no, nucleus is absent. But here if you take young means here definitely which one is present? Nucleus is present. So that is the main point here. If you take young sieve tube element, sieve tube or sieve element, so definitely we can, or sieve tube element, no, definitely we can find out the presence of nucleus. So next one, if you take next one means matured sieve tube element, no, there which one is absent? nucleus is absent. Next one, the functions of sieve tube elements are controlled by the nucleus of companion cells. In sieve tube elements, which one is absent? Nucleus is absent. No, sir, if nucleus is absent at the time, who will do this particular function, sir, or who will regulate the functions of sieve tube elements, sir? Means here the sieve tube elements function, all functions of this one under the control of nucleus of companion cells. Actually, it is super topic, suppose if you are understanding, okay, otherwise definitely in smart classes I will explain by one more time, no worry. Okay, now here in matured sieve tube elements, which one is absent? Nucleus is absent. Even though here in sieve tube elements nucleus is absent, sieve tube elements all works will be under the control of nucleus of companion cells. Next one, the companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. Already this point, so here we have discussed. So here that is a point only I have written. So in the next video, by covering of companion cells, let us finish the information of complete phloem. So no worry of this particular information, definitely each and every line you will learn. So don't worry about all these sentences or new words. So definitely you will learn this particular information. Just now you will learn. Suppose if you are unable to understand, in presence of you, definitely I will explain by one more time in smart classes. Thank you, and be safe at home.